Hello everyone, my name is Andreas Dräger and I have the pleasure to show you today what the Systems Biology Simulation Core Library is and how to use it. So, the Systems Biology Simulation Core Library is a collection of interpreters, solvers and algorithms that help you to deal with systems biology models. You can make calculations and do the preparation for plots in different frameworks. For instance, in stochastic systems, if you want to solve systems with time and get amounts of molecules out, or do constraint-based analyses, which will give you a flux distribution with a certain biomass objective function or other objectives that you might have in your model, and then also ordinary differential equation solvers are included in the Systems Biology Simulation Core Library. So that gives you concentration versus time in an ordinary differential equation framework with continuous equations. So all this is based on a collection of standard data formats in systems biology. Here, most prominently, the systems biology markup language, which can be embedded in a combine archive file and also can be accompanied with a setml file that tells you how to interpret the system. So all this is based on a collection of different tools and third-party libraries. So it's a Java library. So it uses JSBML as a parser and for manipulating the systems, and is also running from the command line if you want. And then to benchmark the algorithms in there, we used models from 3 different collections. So the Cobra, Open Cobra project with CobraPy is one important comparison tool, or also the systems biology markup language has a large collection of benchmark problems, more than 1,000 or so, which is the systems biology test suite. And then Biomodels database with its models provides many different ways to analyze these systems and to see if the solvers work on them. So all this makes the Systems Biology Simulation Core Library a well-tested environment that you can directly embed in your Java application as a computational backend. Here's a usage example to demonstrate what it does. So let's assume you have a little system here, in this case 2 reaction equations, which can be displayed graphically if you like here, and then you want to solve that system. So first of all, you would have to load the SBML model here with the SBML reader, for instance, that comes from JSBML, and then you would initialize an interpreter for this system, in this case the SBML interpreter here, and that internally constructs a differential equation system with a stoichiometric matrix and the flux vector that you don't have to deal with anymore. And then you can solve the system with any differential equation solver, in this case here the Rosenbrock solver, and then set also the step size if you want, for instance 0.3 or whatever you find appropriate for your system. Then you obtain the initial values that should be included in the SBML model already, so the interpreter makes some computations to get those initial values from the model, and then you can solve. So to this end you simply say solution equals solver.solve, and you give them all these variables that are needed, end time and start time, and it returns you a table with values of concentrations and also flux values that can then be the basis for plotting the result, for instance here like this, in your favorite plotting environment. To embed SBSCL, the Systems Biology Simulation Core Library, in your Java application, it is most convenient to use Maven. Here's a minimal example how to do that. So first of all, you have to declare in an XML file, in the POM file, the basic settings for Maven that you always have to do, and then you declare the details of your program. It's recommended to set some variables also that you can reuse, for instance how the date format is, or what building environment to use, and some other properties. And then the most important part is displayed here. By entering this little fragment of code, you will directly have access to the Systems Biology Simulation Core library in your application. Maven will automatically download this software, and you don't have to care for anything. Finally, you also have to define how to build your project, and with these, like 55 or so lines of XML code, you can already run your application with SBSCL as a solver backend. I like to thank you for listening, and thank all the people who contributed to this effort. Also thank the funding agencies, in particular the Google Summer of Code project. And if you're more interested in our efforts here in Systems Biology, just read the preprint that is already available, use the DOI that you can see here, or watch more videos on our YouTube channel. Thanks, bye bye.